Hello. Today I'll be explaining Punnett squares and showing you how to use them. Punnett squares are tools used to make finding the possible offspring of two organisms easier. There are many instances where Punnett squares could be used in our lives. One of the main ones is with gametes. Gametes are the specialized cells involved in sexual reproduction, so the sperm and egg cells. Even though these might be the most relevant to an average person, I will be talking about crossing two pea plants. This Punnett square will show the possible offspring of a homozygous yellow plant and a heterozygous yellow plant. So you can see that there are these G's up here. One of those G's is a gene, so this is what the gene would normally look like. That GG, that would be the gene. Then one of these G's, so just this here, that's considered the allele. That's just one of the traits in the gene. Uh, the ones on the top, you can see they're both uppercase. That represents them both being dominant. Dominant means it's always shown when, like, it has that dominant gene. So, or dominant allele. So you can see on the left, it has a dominant, which is uppercase, and a lowercase, which is recessive. In this case, the uppercase is yellow and the lowercase is green. So since it has the dominant trait, which is the uppercase, that would make the plant yellow because it has the dominant, so it's always being shown. The only case where you would see a green pea plant is if they were both lowercase g's. That would make it a homozygous recessive plant, which would make it green. Homozygous is when they're both the same, like uppercase or lowercase. So this one up top would be homozygous, and then heterozygous is when there's an uppercase and a lowercase. This one on the side would be heterozygous. Then to make your Punnett square, you want to move this top G, you want to move it down the row. So you would start here, make that G, then this G here. Then you do the same with the other G on the top. So you'd go G here and G here. Then you would start moving across. So you'd make this G here go over here and then over here. Then you do the same with the lowercase. So it'd go here and here. This is what gives you the genotype ratios. So you can see that these GGs are the genotypes. So your genotype ratio for this would be 50% GG, and then 50, wait, let me get to a draw. Then 50% GG. Then that gives you the genotypes. That's the gene it shows. That's the gene and the ratio of that. The phenotype is what is shown in the plant. So all of these, as you see, they have an uppercase G, so they have a dominant trait, which would mean they're all yellow. So the phenotype would be 100% yellow. And that's kind of the basics on Punnett squares. You just need to remember the dominant and recessive. And when there's a dominant, it will always show up.